In this lecture, we're going all the way back down towards the bottom of this pyramid in our levels of organization that we went through, I think, in our first lecture. So I didn't draw the whole thing again. I just kind of started with the very bottom layer of atoms. And we put some examples last time of individual atoms. I know there's no periodic table of elements in here, but there is a portion of a periodic table on the second page of your lecture handouts. It at least shows the individual elements that we talk a lot about in biology. Some of those will include, some of the more common ones will include carbon, oxygen, hydrogen, and nitrogen. And we'll talk about the chemical bonds that allow us to stick those atoms together to build molecules. And some examples of molecules we can see in the level above. I've drawn a carbon dioxide molecule, or an oxygen molecule, or a water molecule, or a methane molecule. We're going to describe those molecules as either being polar or nonpolar. I know we don't have anything up yet about polar versus nonpolar, but that's going to determine how they behave in the level of molecular bonding. This is pretty much all we go through in chapter 2 and 3. We're going to spend the rest of the time in the molecular level of organization. In Unit 2, we get into organelles and cells. So just kind of a quick roadmap of where we are. The past couple of lectures, we were talking about big-scale concepts, evolution and things like that, which if you've been thinking about it and you have questions, certainly bring those back up. I love talking about that stuff. But where we are going in this lecture, we have to get down to some of the nuts and bolts of basic chemistry from the perspective of biology. Because one of the first things that I wanted to write on the board, like I said, was why are we going down to the chemical level in the first place? Let's visually look at why this is relevant. And maybe I can introduce these terms polar versus non-polar before we get too far. Like I said, it all comes down to trying to figure out the rules that allow things to get into a plasma membrane. And I'm going to draw, let's just put a sample plasma membrane. I'll try and make it so that the screen doesn't block us out. I'm going to put a row of molecules that have a rounded head and these two twisty little tails. And these molecules, to look at just one of these molecules all by itself, we're going to call this a phospholipid. Hopefully that's not too faint that it's difficult to see. This phospholipid, let me switch colors in case it is separated into this round portion that's described as the head of the phospholipid. And then you have these two tails of the phospholipid. What we can remember about those is the head of the phospholipid is going to be polar. We need to define what polar means. The head is polar. The tails are going to be non-polar. Tails are non-polar. Let's see where that's going to come back. When we draw the rest of this plasma membrane, we're going to see that each plasma membrane is made of a double layer of these phospholipids. So I've got to put these tails that are non-polar facing the other row of tails that are also non-polar. You can see the heads are facing opposite directions. Polar molecules, we will learn, they like to be touching other polar molecules. Now that we've got our plasma membrane in here, let's just label here's our plasma membrane. Let's label the top side of the board. This is going to represent the fluid outside of the cell, our extracellular fluid. Extracellular fluid versus the fluid on the other side of this plasma membrane inside the cell. We're going to call this the intracellular fluid. Intracellular fluid is also referred to as the cytoplasm. I thought I had one of these that was not running out. Let's try this one. What's important about this plasma membrane is it separates the things outside of the cell from the things inside of the cell. And to use maybe a weird analogy, this is always used, I don't know why, it goes all the way back to Camelot days, but this plasma membrane is sometimes described as the moat on the outside of the cell that protects the inside of the cell, makes it difficult for things to just move into the cell. And this, we have to imagine a time when there were castles, and to protect the castle, they would dig these moats and they would fill it with alligators and things like that. That was a deterrent, making it difficult for people to move into this castle. There aren't alligators in the plasma membrane, but there is something in the middle of this plasma membrane that makes it difficult to cross. And that the alligators basically are this double layer of phospholipid tails. It makes a very thick wall of non-polarity. So I'm going to describe this as a non-polar wall. You've got this thick layer of a non-polar barrier or moat. It's going to make it difficult for things that are polar to cross. Non-polar things are going to be able to move across the plasma membrane easily because the entire membrane is made out of this non-polar barrier. I should say the extracellular fluid is going to have lots of water outside of the cell, and even the cytoplasm inside the cell is going to have a fair amount of water in it. And these polar heads, they prefer to face wherever there's a water molecule. The tails, like we said, they shy away from water molecules, so they face inwards towards one another. And they form this nice effective seal. We're going to see that anything that is outside of this wall, that is too big, like this glucose molecule, this is too large to be able to cross that plasma membrane anywhere. It's prevented from crossing. We're going to see there are two criteria that determine whether or not a substance can cross the plasma membrane easily. I'll just list those two. The two criteria are size and charge. So the whole reason we're talking about molecules having a slight charge or not is going to play a big role in how easily they can cross the membrane. If you're tiny enough, if you're small, you're going to be able to move across the plasma membrane. Water molecules, we will see, are small enough that they can just wiggle through the plasma membrane. This movement of water molecules across a membrane is going to be what we describe as osmosis. But in addition to water crossing the membrane, we will see that there are additional proteins. We're going to end up spending a lot of time this semester talking about these proteins that are embedded in the plasma membrane that allow particular substances to move through. This one I'll abbreviate as AQP. It stands for aquaporin, and there's like 20 different types of aquaporin proteins. They're like a hollow straw that when stuck onto the plasma membrane, they make it easy for vast quantities of water to move across the membrane. Normally it's a slow process of water to wiggle through, but aquaporins allow rapid movement of water into the cell. In fact, your immune system can kill off certain bacteria by moving into these bacteria and putting a bunch of these aquaporins all over the bacteria. Now it's taken on more water than it can handle, and that causes the cell to implode. So aquaporins are important. In addition to aquaporins, there are specific proteins that allow large things like glucose to move across the plasma membrane, where normally its size would have excluded it. We're going to call these GLUT proteins. It just stands for glucose transporting protein. And then finally, we're going to see, we said the charge. If any of these tiny particles like an ion have a charge to it, whether it's a positive or a negative charge, we'll say potassium or one of these hydrogen ions, if there's a charge to this ion, it's not going to be able to cross freely on its own. It's instead going to rely on another one of these proteins. Let's put a phospholipid in between there. These are going to be proteins that are specific to individual ions. We'll see transporters that are specific to sodium or potassium or hydrogen ions. So again, plasma membranes are going to be highly selective in what is allowed to cross. Most of the substances are going to be crossing selectively through particular protein channels, whereas the plasma membrane itself is going to be this barrier of non-polarness. Now, we still haven't talked about what polar or nonpolar means, but we know that the plasma membrane is basically made out of this nonpolar wall. And, well, we'll explain why that's easy to cross for other nonpolar things as we get into that. But at least we've kind of gone all the way backwards to explain as we get into chemistry. What we're looking for is how do we build these molecules and what determines whether these molecules are polar or nonpolar, how easily they cross the plasma membrane. Now, to the slides. This starts way back at the beginning where we are reminded, like in a physics class, that all organisms that we study in here are composed of matter, and matter exists on this spectrum of solid, liquid, and gas, where solid is a state of matter where the atoms are typically more closely packed together and it's more dense. Whereas in the gaseous state, these atoms are spread far apart from one another and rarely interact, so it's much less dense. Somewhere in between solid and gas, you've got liquid. Most of the time. There are some unique characteristics about water that we will talk about. It's one of what is unique about water, one of the many things that's unique about water, is that it is one of the only substances that is less dense in its solid state. It's actually more dense in its liquid state. What's going on? We'll talk about this water molecule once we build a water molecule. There are 92 naturally occurring elements. We don't have the periodic table of elements in here, but we do have that simplified chart that, like we said, is on the second page of the lecture handout. It goes through most of the atoms that we will encounter here in a biology class. I keep throwing around this word atom. Remember, atom is the smallest unit of matter that still retains the properties of an individual element. Each element is going to have its own atomic number, the number of protons. We're going to see, like I said, the majority of atoms that we focus on in here, I've erased it from the board, were carbon, hydrogen, oxygen, and nitrogen. Those are the main atoms that you find in living organisms. There are a few other ones in trace amounts, like potassium and sodium and calcium. Phosphorus is in there, to name a few. But we'll focus mainly on the interaction between carbon, hydrogen, oxygen, and nitrogen. And when kind of investigating the chemical bonds that these types of atoms like to form, we have to first look at the atomic nucleus. And this is fascinating. If this was a physics class, I feel like we would spend weeks on just this slide alone, because it shows a comparison of an atomic nucleus compared to its electron cloud. The atomic nucleus is where you would find the the atomic nucleus is where you would find the protons and the neutrons. They're right there in the middle. And around this nucleus of protons and neutrons, you have the electron cloud. It behaves more like this image is showing us. It's something that exists in a cloud of probability rather than how it's typically drawn, like these electrons are orbiting around the nucleus, kind of like the moon orbits around the Earth. It's easier to draw them this way. I'll draw them like that on the board. But in reality, these electrons don't behave that way. They behave more like waves. And what's so fascinating about that, and why I would spend weeks on that in a physics class, is because there have been experiments really done in the last decade that show how these electrons behave more like a wave than a particle. And I always thought it was a fascinating discussion to think about, are things particles or are they waves? Turns out electrons behave more like a wave. It's really interesting. Not that we have to go that deep. What we need to know is protons and neutrons are in the nucleus. Protons always have a positive charge. Neutrons are neutral. They have no charge at all. The negative charge comes from those electrons that exist in this cloud of probability. Has anybody seen those double slit experiments where they pass these electrons through these double slits and you get these interference waves on the other side? Not that we have to see it. Maybe YouTube it when you leave here if you're interested in how these things indicate that electrons behave like waves on the surface of a water, smooth ripples on the surface of smooth water, basically. Anyway, this electron cloud will draw as they, in what would appear as if they were orbiting like moons around a planet, but this is just an oversimplification to allow us to write them on the board. So we'll get over that. When we look inside the nucleus, there are the protons and the neutrons. The atomic number is going to be unique for each individual element. This atomic number, atomic number at the top, is referring to just the number of protons that an element has. And that's unique, like we said, for each individual element. If you were to change the number of protons, well, now we're talking about a totally different element. So proton number doesn't vary without changing the number or changing the element that you're talking about. Neutrons, those neutral particles in the nucleus of an atom, have no charge. They do have mass. So if we wanted to calculate the total mass of each of these subatomic particles, the mass is measured in daltons. And a proton would measure in one dalton, a neutron would measure 1 dalton, and electrons are much smaller. They have really a fraction of mass. So the atomic mass is going to take into account protons, neutrons, and electrons. Mass number is just an indication of protons and neutrons. This, it looks like it has 6 protons, so automatically we know we're talking about the element of carbon. Atomic number, like we said, is just the number of protons. Mass number is telling you the number of protons and neutrons. And atomic mass is the combination of protons, neutrons, and electrons. So back to this, we said proton number doesn't vary. What I like about this picture is it shows we've got 3 different versions of a carbon atom. In all 3 versions of a carbon atom, we see 6 protons. That never varies from one individual element to the next. So we've got 3 atoms, all of the same element, which is carbon. But we can see what varies in these is the number of neutrons. This varying number of neutrons is what leads to different versions of carbon. We would refer to these 2 different versions of carbon as isotopes of carbon. An isotope, we can see these latter 2, as they grow larger, like carbon-14 is larger than carbon-13, they become bulkier and more difficult to hold together. They become less stable. So these bulkier isotopes can fall apart at a fixed rate. And this decay or this falling apart of these isotopes is what's called radioactivity. So these radiometric isotopes decay at a fixed rate. And that's useful for biological purposes. In your class notes, it wants you to list a couple of things that these radiometric isotopes are useful for. And one thing they are useful for is in chemotherapy. So if you're taking chemicals that need to target certain cells and then radiate to kill off certain cells, then chemotherapy is one application for these radiometric materials. And then another one is dating, using these radioactive isotopes to do radiometric dating and tell basically how old certain... What's interesting is the half-life of carbon might take us back maybe to about 10,000 years ago or so. And if we want to date things older than that, we need to use different isotopes, some other isotope other than carbon. You can use uranium. There's several isotopes that go way, way further back than how far back carbon will date. But anyway, we're talking about isotopes. These are different versions of an element. Their proton number never varies, but what does vary is the neutron number. The larger they are, the less stable they are, and the more radioactive they become. Here's the picture that you have in your class notes. And what I like about this one is that it shows kind of the Bohr diagram of each one of these elements. It's just giving you a big dot to represent the nucleus where the protons and neutrons would be, and it's kind of simplified where we would find those electrons. It's drawing them like they are orbiting around the nucleus. But it's a quick check to see how many electrons are present in the outer shell of each one of these elements. Where that is important is, like I said, we don't have to know all the elements in the periodic table, but we have to know at least the first 3 rows. So here are some guidelines that will allow us to quickly reference whether these individual elements are highly reactive, unstable, looking to make some kind of deal, or whether they're totally fine, not looking to interact with anything. Is it okay if I erase our diagram about the plasma membrane? Not that you need to really remember that, other than the fact that we're going to be talking about polar and nonpolar. Nonpolar can get through a membrane. Let's practice with some elements real quick, and it will help explain why when I was drawing things like sodium or potassium or calcium, we'll draw potassium like this, or sodium with 1 positive charge. I'll draw calcium with 2 positive charges. So what's going on there? How do these individual elements in the periodic table end up with a positive charge or a negative charge? It has to do with these rules of chemistry, and I'm just going to go over here in the corner, and instead of saying sodium, potassium, or calcium, I'm just going to put an X to be a general representation of any element in the periodic table that we'll talk about in here. What we need to remember is that surrounding the nucleus of each atom, there are going to be these little shells or orbits that house electrons. And the first little shell or orbit around a nucleus is small. It can only hold 2 of these electrons. Instead of drawing an electron, I'm just going to put the number 2 to show us 2 electrons can fit in that first shell. And then as we move out, these shells get a little bit larger and they can accommodate more electrons. The second shell is large enough that it can hold 8 electrons. And the third shell is also large enough that it too, if I can get that in there, holds 8 electrons. And for the purposes of this class, that's as far out as we need to go. We can remember the first shell is going to hold 2 electrons. The next 2 are going to hold 8 electrons. The additional thing to remember with this is that atoms in the periodic table, they want to have their outer shell of electrons completely full. We'll say instead of saying needs, I'll say outer shell wants to be full. It wants to be full, and if it's not, it's going to be looking to make some kind of deal. If you grab one of these elements over here, like neon, for example, it has an atomic number of what looks like, what is that, 10? I can't read because I'm too close to that. Yeah, so neon has an atomic number of 10. That tells us 10 protons, 10 electrons. If we wanted to do this diagram where we draw the first shell, 10 protons, 10 electrons. We know the first shell is big enough that it can hold 2 electrons, so I'm going to put 2 electrons in there. I've got 8 more to pass out, so that takes us out to this second shell. And we know the second shell can hold up to 8, and that's exactly how many I have to pass out. So that outer shell is completely full, and that makes neon one of these noble gases. We can't get it to react with anything because it's already happy. It's content. Maybe happy and content are really not the right words. What we're saying there is that it's stable. Its outer shell of electrons are completely full. Let's look at something that doesn't have an outer shell of electrons that is completely full. I'm picking on this sodium now. Let's grab this sodium. It's got an atomic number of 11. Where are we at? Here it is. So that tells us 11 protons and 11 electrons as it comes straight off the periodic table. So let's pass out those 11 electrons. We know the first shell is big enough to hold 2. Let me get some of this out. First shell, we can fit 2 of these electrons in there. 1, 2. Now we get to the second shell. It can hold 8. We've got 9 to pass out, so we quickly fill up. Quickly fill up that second shell. We've only got 1 more to pass out, and we have to go out to this third level to do it. We know this third level wants to house as many as 8 electrons, but we've only got 1 to put in there. So sodium is not stable. It's looking to make some deals. And it turns out that it would be more difficult for sodium to pick up 7 of these electrons and fill the entire outer shell. That's 7 steps, gaining an electron each time, as opposed to just doing the one step of kicking off its outer electron, donating it, getting rid of it, losing it altogether. Now, if it gets rid of that outer electron, we can erase this entire outer shell. But we have to go back here and do the math. One more time. We've now lost an electron, so we have to say there's only 10 electrons. It's uneven now between electrons and protons. There are more positive charges than there are negative charges. In fact, 1 more positive charge than there are negative charges. Now that this electron has been cast out, we have formed a positive ion, one of these cation ions. Let's look at the other one on the other side of the table. Anything in this first column, you can see hydrogen, lithium, sodium, even potassium down here, would all have 1 electron in its outer shell. Much easier for these to kick off their electron rather than to pick up another one. Magnesium, you'll look, it has 2 electrons in its outer shell. It's easier for magnesium to get rid of those 2 electrons, so sometimes magnesium is drawn like this, with 2 pluses, just like calcium, because it's gotten rid of 2 of those electrons and it now has 2 more protons than it has electrons. So I'm so OCD that I have to write 2 positive charges next to it.
So the protons and electrons are not always equal, because I thought they were always the same.
So the number of protons from one element to the next doesn't vary. If you change the proton number, then you change the element that you're talking about. But what we will learn is that some of these elements are unstable because their outer shell of electrons are not completely full. So in order to become stabilized, they're going to have to make some deals with their electrons. And that's what throws off the number. Sometimes they lose electrons, and that casts them as a positively charged ion. In this other far right column, we're going to exclude the noble gases because, like we can see, they're not interested in making deals with anybody. They're fine. But in this 7th column, if we look at something like chlorine, it's got an atomic number, we'll just stick with this, it's got an atomic number of 17. So that tells us initially it starts out with 17 protons and 17 electrons. So it doesn't have a charge yet, but it's not stable. It's going to be looking to make some deals. So as soon as you find it in nature, we're going to see it's in this situation where, let's draw these 2 electrons in the first shell. We get out to the second shell. We know 8 can fit in there. So 1, 2, 3, 4, 7, 8. We've got 7 more to pass out, so that takes us out to this third shell that can hold 8, and we've only got 7. So 1, 2, 3, 4, 5, 6, and 7. This thing is unstable. It's looking to make some deals. It wouldn't just exist on its own. And one of the things that it does to become stable, it's got 7 already, so it's easier for it to go through the one step of just picking up an extra electron that might be floating around in the environment. It would be easier to do that, like we said, than lose 7 electrons. So we typically see chloride gain one of these electrons, and as soon as it picks up an electron, its outer shell is stable. It's now happy, but we have to go back and do our math. That shows us we've now got more electrons than protons, more negative charges than positive charges. So this chlorine, after it's picked up its electron, we mark it with a negative charge. And just like the two ends of a magnet, we've got opposite charges that are attracted to one another. So this is going to be a type of bond that will end up forming molecules like salt. Sodium is a reactive metal. Chlorine is a toxic gas. But when we combine these two, we get table salt, something that you have to have in order to survive. Chlorine gas on its own would kill you. Ingesting sodium on its own would kill you. But again, combining these two things gives us a totally new molecule that we actually need. And that's something that's different about these chemical bonds versus molecular bonds. We'll see that chemical bonds actually make something new. They combine elements, and the combination of these elements make a molecule that have totally different properties than the individual elements themselves. So chemical bonds are going to make something new, whereas molecular bonds do not make anything new. All we are talking about at the molecular bond level are how these molecules interact with one another. Do they like one another or not? I was talking about chemical bonds making something new, and molecular bonds don't make anything new. So let me say this. If we catch up with our slides, I had written our rule of 8. The first shell holds 2 electrons. The next 2 shells are going to hold as many as 8 electrons. And the magic, like we were saying about what makes these individual elements want to grab an electron or get rid of an electron, has everything to do with that the outer shell of each individual atom, in order for that atom to be stable, needs to have the maximum number of electrons, needs to be full of electrons. The noble gases, like we said on the far right, are already completely full of electrons, so they are stable. They don't want to react with anything. Here are the generalizations that we're going to make about the periodic table of elements. Periodic table of elements, if you're keeping track, there's one verbal mutation so far today. What I've highlighted in green here are the columns to the far left or right of this periodic table of elements. These are the ones kind of at the edges of the periodic table. They're in the boat that sodium and chloride are in. The ones that are on this side of the table, they've got either 1 or 2 electrons in their outer shell. It's easier for them to give up those electrons than, get rid of 2 electrons than it would be to gain 6 electrons. On the opposite end of this periodic table of elements, you have things like chloride that have 7 electrons in their outer shell, and it would be easier for them to gain an electron than it would be for them to get rid of a bunch of electrons. So it makes sense that on the edges of the periodic table we see a lot of this gaining and losing of electrons. But the elements that are in the middle, it's not easy for them to gain or lose as many electrons. They're somewhere in between. And to give us an example of that, let's make a little space. That might be just enough space to investigate carbon. Is there a question that I missed? If my head is turned, just yell at me. Don't hesitate to yell at me. I'm going to write carbon. Hopefully this is dark enough that we can see it. Carbon has an atomic number of 6, so we've got 6 protons and 6 electrons. When we go to write our little shells around this, we know there are 2 electrons that we can fit in that first shell. 1 and 2. And now we've got the second shell that can hold 8. But we've only got 4 to put out here. So we're kind of right in the middle of the road where carbon, it's not easy for it to gain 4 electrons. That's 4 steps. It's not easy for it to lose 4 electrons. That's also 4 steps. So carbon's in the middle. It doesn't tend to gain or lose electrons. What do you think carbon does? Where are we going with that? That's right. I heard it. It shares those electrons. We're narrowing in on polar versus nonpolar. are two types of chemical bonds. We're going to see the edges of the periodic table. They tend to either lose or gain electrons. Any bond, let me say that again. Any ionic bond is going to be between molecules that have either gained or lost electrons. So let's describe ionic bonds as a gain or a loss of electrons. They have to do with a gain or loss of electrons. And that creates those positive ions or negatively charged ions. It's always a bond between two oppositely charged ions. Whereas a covalent bond is not due to gaining or losing electrons at all. Like was just mentioned, covalent bonds are a situation where there is sharing of electrons. Sharing of electrons is much more of an intimate type of bond. We've got carbon. It's unstable as it sits. It's looking to get access to at least 4 more of those electrons. I'm going to steal some space away from this for a second. We're kind of crowded in here, but look what happens if we draw off to the side an individual hydrogen. It's got an atomic number of 1, so it's got 1 electron in its first shell. That's not enough. It wants access to at least 1 other electron, so this hydrogen all by itself is unstable. If we can pull this hydrogen closer to the carbon, close enough that it can bring its 1 electron and share it with the carbon. Let's draw this hydrogen as it moves in a little bit close. With its 1 electron, it's now brought close enough to this carbon. As long as it's sharing 1 electron with this carbon, it's held in very close proximity. That gives the carbon access to 1 additional electron. Let's imagine if we brought in, and here's where I have just enough room to squeeze in another hydrogen. Each hydrogen is bringing its single electron with it. And now as we draw these orbitals around each hydrogen, we can see each individual hydrogen is content or happy because each hydrogen has access to 2 electrons. It's now stable. And if we look from the perspective of carbon, its outer shell now has access to 8 electrons. It's now stable. The molecule that we have formed is CH4, or methane gas. This methane is going to be what we will call a stable nonpolar molecule. Let's write nonpolar. That's a nonpolar molecule. Let's compare something that is polar. Polar versus nonpolar. We now have walked into that discussion. So let's add to this real quick. Covalent bonds have to do with sharing of electrons. And anytime you're sharing anything, whether it's electrons or money, there's 2 ways that you can share that. There is either going to be equal sharing or unequal sharing. Equal sharing is going to be tied to what we will describe, let's get something bright. Equal sharing is going to lead to molecules being nonpolar. Unequal sharing is going to lead to molecules being polarized. What is going on there? Let me use this analogy. Imagine that I brought a big treasure chest into the middle of this classroom. It's sitting right there in the middle of the classroom, some big treasure chest, and we open that treasure chest up, and it's full of stacks of $100 bills, crisp ones, never even been bent. What would happen then, if we were going to try and make this room non-polarized, I would ask everybody to spread out around the perimeter of the room so that we're all equally spaced around the perimeter of the room. And then if I took out those stacks of $100 bills and I equally shared them amongst everybody, so everybody got 1 stack of $100 bills, we would have a room that is non-polarized. Everybody would be equally happy on any side of the room because we all got the same amount of money. We're sharing this equally. But in order to polarize the room, let's imagine I open up that treasure chest of $100 bills and I give one side of the room no money at all, and then the other side of the room, everybody gets 2 stacks of $100 bills. These people over here are going to be really happy, but they over there are going to be pretty mad they didn't get any money at all. So when we are not sharing things equally, it leads to polarization. Molecules can be polarized if they are not sharing electrons equally. Now that we understand how chemical bonds play a role in determining whether a molecule is polar or nonpolar, let's carry these words down here to talk about molecular bonds. We have 2 types of molecular bonds. Neither one of them are going to make a new molecule. They're just going to hold certain molecules in close proximity. And it turns out hydrogen bonds are these types of weak bonds between polar molecules. So we'll say bonds between polar molecules. So hydrogen bonds are what hold these polar molecules next to one another. And it turns out there is another type of bond between nonpolar molecules. We call it hydrophobic interactions. And I think maybe interactions is a better way to describe these because it's not like a true chemical bond where we're making a new molecule. It's an interaction that holds molecules close together. And hydrophobic interactions are going to be these bonds that hold nonpolar molecules together. Let me use an example, a visual one. I grabbed a water bottle from my truck and I ripped a label off of it, but proven to you that it's water. I guess I won't pass this around, you'll take my word for it. We've got water in here and we're going to see whether these water molecules are polar or nonpolar. We're going to draw this on the board, but let's just see if we can make this work. I'll try and set this up so that you can see. Maybe this is better. Yeah, that's a little better. First thing I'm going to do is, so that we can see that water, I'm going to hit it with a drop of food coloring. Just one will be enough. We can see in a quick little swirl that food coloring easily mixes through the water. Let me draw what's going on first before I add the next step of that. And markers. Let's go over here, and instead of drawing a methane molecule, let's look at what happens if we take, there we go, one of these oxygen molecules out by itself. Oxygen has an atomic number of 8. So we've got 8 protons, we've got 8 electrons. When we go the route of writing out carbon, when we write out the electron shells around oxygen, once again the first shell holds 2 of those electrons. The second shell can hold as many as 8, but we've only got 6 to pass out. So here's how I'm going to spread them out in oxygen. I'll put 1, 2 there, 3, 4, and then down here we'll just go 5 and 6. So the outer shell is not completely full. It's missing 2 of these electrons. One of the ways that it can get access to 2 of these electrons is by hydrogen kind of bringing over its single electron. I'm going to bring, let's just pull this hydrogen molecule as kind of coming in down here, bringing in its 1 electron. There's its 1 electron, and it's now in close enough proximity to this oxygen that it has access to one of those oxygen's electrons. So this individual hydrogen is content. Here's the second hydrogen that comes in with its own electron. And as long as it's in close enough proximity to this oxygen, it too is happy. And now that the oxygen has access to two of these hydrogens, the oxygen itself is now content and happy. It's got access to 8 electrons in its outer shell. But what we have just built is this water molecule, which we will learn is a polar molecule. And what we mean by that is, if I can just put a box, let's just put a box around this water molecule. We can see that one end of this molecule is the end in which we find all of these electrons. For example, the opposite end of this molecule, these two bare protons are what we see exposed at this end. Protons have a positive charge. Electrons have a negative charge, and most of those electrons are at this end of the molecule. So we can mark polar molecules as having one end being slightly negatively charged and the other end of the molecules being slightly positive charged. Each polar molecule is like a little magnet in that sense, and that's why when I put the food coloring into the water molecules, the food coloring is also made out of polar molecules, and that's why they snapped together so quickly. Look, let me put another one of these. Instead of drawing the entire hydrogen, sometimes we're going to draw water molecules just like this, with the oxygen and 2 hydrogens attached to sticks. But now that we know this is a polar molecule, we have to imagine that at the hydrogen ends there are these slight positive charges, and at the oxygen ends there's a slight negative charge. Let's put a few of those down here. If I had another one with an oxygen and 2 hydrogens down here, the hydrogens, there are always going to be positive charges associated with them, and the oxygen, there's always a negative charge associated with it. We can even put little boxes around each of these water molecules. And we said a hydrogen bond, I'll represent it with this green arrow, it is just a weak bond between the negative end of one polar molecule and the positive end of another. This is our hydrogen bond. Notice that we still have 2 water molecules. This doesn't change the properties of what those molecules are. It's just a bond that holds them in close proximity. So all of those water molecules are held together like these tiny little magnets. Maybe the thing that might be helpful in determining quickly whether a molecule is a polar molecule or a nonpolar molecule is this little, I don't know, it's not like a cheat sheet or anything, but it is a helpful chart that allows us to look at a molecule and tell whether it's going to be polar or nonpolar. This sharing of electrons, kind of how hard the nucleus of an atom can pull on electrons, is measured in chemistry by what's called electronegativity. Electronegativity is, like we said, the pull of those electrons by any atomic nucleus. And the ones that we compare mostly, we're going to see that if we compare carbon to a hydrogen atom, they both have the same strength in terms of electronegativity. They're going to pull equally as hard on those electrons. So in my brain, I like to imagine a tug-of-war game. And on one side of the rope, you've got a bunch of carbons. And on the opposite side of the rope, you've got a bunch of hydrogens. And if they're the same strength, then that rope isn't moving anywhere. It's just staying in the same place. They're equally as strong. As opposed to if we compared carbon or hydrogen to those other 2 common atoms that we see a lot, oxygen and nitrogen. Oxygen and nitrogen are going to be much more electronegative than carbon or hydrogen are going to be. So, for example, in a water molecule, we have a hydrogen and an oxygen. And the oxygen is stronger at pulling on those electrons. So in this tug-of-war, the rope with all the electrons comes over to the oxygen side. And so now this side of the room with all of the oxygen, let's try that again. This side of the room that has pulled that electron rope over here has all of the electrons. So this side of the room will be negatively charged. And that side of the room with the lack of electrons will then become positively charged. That That works for just about everything except there's one exception, and let me put that on the board real quick. That is this molecule of carbon dioxide. If you just look at that, you would think, well, we've got carbon and oxygen, so clearly the oxygen is going to pull harder on those electrons than the carbon would be. And that is true, but when we actually draw this molecule out, it turns out that carbon is kind of stuck in the middle, and it's being pulled on either side by an oxygen that's formed this double bond with each carbon. So carbon isn't going to be able to pull on those electrons equally as hard. It's just that it's kind of stuck in the middle. It's the rope, and then it's being tugged on either side by these oxygens, so carbon's just kind of stuck in the middle. Carbon dioxide, because of its atomic structure, there is no one side where there are more electrons than the other. So this too would be a nonpolar molecule. All nonpolar molecules are capable of forming these hydrophobic interactions that hold them together. I did find some nonpolar molecules just before I left the house. There is some vegetable oil that I have not yet used to fry an egg. So I brought it in here, and these are all nonpolar molecules. So let's see what happens when we put nonpolar molecules together with polar molecules. If these bonds work, the hydrophobic interactions should keep this vegetable oil from mixing with the water and the food coloring. Let's see how this works out. I'm just going to set it over there, and it should take just a couple of minutes. Doesn't look like they're wanting to mix at all, and if we give that just a second to settle, we should see those completely separate. So hydrophobic interactions are holding together all of the nonpolar molecules. We can see them adjusting, and all of the hydrogen bonds are holding together the polar molecules at the bottom. I forgot to take that out of a classroom at another campus that I taught at, and the cleaning crew came in there and thought it was a bomb. And I was named the Unabomber of that campus. But I guess they saw Die Hard 3 or something and thought that was like a bomb. But it's not. It's not going to mix together because of one of those molecules being polar and the other one being nonpolar. Okay, so anyway, let's get away from this slide. We said elements that are on the edges of this periodic table, they tend to gain or lose electrons. So they form these ions, positive or negative ions, and those ions can lock together to form these ionic bonds. Atoms that are in the middle of this periodic table of elements, like carbon was, it wasn't easy for carbon to gain or lose 4 electrons, so it did the opposite. It shared electrons with other atoms. And that took us into this world of covalent bonds, or sharing electrons. In the world of sharing electrons, this is what leads to polar or nonpolar, equal sharing or unequal sharing. Now that we know if molecules are held together by covalent bonds, and that bond is not equally sharing electrons, that's a molecule that's capable of forming hydrogen bonds. Nonpolar molecules, like we said, are capable of forming those hydrophobic interactions. Said that a bunch. Let's see how we're doing on time. I got about 15 minutes before we take a break. So let's catch up with some slides. We have listed ionic bonds and covalent bonds. This is just a picture that shows what I attempted to draw on the board, that sodium, as it comes out of the periodic table of elements, it's neutral. It doesn't have a positive charge yet, but it's unstable. It's not going to stay this way long. In order for it to become stable, its outer shell of electrons needs to be full, and we said it's easier for sodium to kick off an electron. So after it's done that, we now have to refer to sodium with a positive ion, indicating that it has kicked off one of those electrons. Chlorine is on the other end of the periodic table. It's got 7 electrons in its outer shell, so much easier for it to pick up an extra electron. And when it does, we have to redo our math and put that negative charge next to this ion, because the negatively charged electrons outnumber the positively charged protons. So those are ionic bonds. Table salt is a common example of an ionic bond. I should say that in biology, we consider these ionic bonds to be weaker than covalent bonds. If you take a chemistry class, they might tell you something different. But in biology, we're talking about chemical reactions that occur in solution, in cells, basically. And cells have the consistency of, at least the first cells have the consistency of ocean water. So there is an aqueous solution in all cells. So all of the chemical reactions that take place in life have to take place in liquid water. Liquid water, because each water molecule is like a little magnet, when we submerge something like sodium chloride in water, I don't know what's going on there, when we submerge sodium chloride in water, these positive ends of the water molecule, the hydrogen ends of the water molecule, are going to completely surround this negatively charged chloride and separate it from the positively charged sodium ion. All of the negative ends of the water molecule, the oxygens, will surround the sodium, and the two will be separated from one another. So ionic bonds will dissociate from one another when submerged in water. Covalent bonds won't do that. If you're talking about plastic and metals, things that will not be submerged in a human cell, in those situations, if you're not going to bury it in water, ionic bonds might be stronger than covalent bonds. So really just in the perspective of biology will we consider ionic bonds weaker than covalent bonds. Covalent bonds, we said, are more of an intimate bond where these orbits of electrons have to overlap one another. If we had two hydrogen molecules there would be a tug of war. All right, let's just put one of those electrons between two hydrogens. Those hydrogens are equally as strong, so this hydrogen gas molecule would this be polar or nonpolar? What did I hear? Nonpolar. It's equally sharing those electrons. Two hydrogens pulling on this rope of electrons. They're the same strength, so there's not, the rope isn't going to go to one side or the other. All right. If it's unequal sharing, that leads to polar molecules. In looking at this picture, we can see hydrogen gas equally sharing electrons. We can see two oxygens equally sharing those electrons. We have drawn up here one of these methane molecules where, if we were to go to any side of that molecule, we would see the same number of electrons, just like our analogy of equally sharing those $100 bills. If those $100 bills were spread equally across the room, everybody would be equally happy. But down here, we can see those oxygens hogging those electrons. So there is, in a water molecule, one end that we can designate as being slightly negative, whereas the other ends are going to be slightly positive. It's drawn like this in the slides. In fact, looking at this water molecule that's polar, we can compare it to our methane molecule that is nonpolar. We said hydrogen bonds can happen between any polar molecule. So this is a water molecule that's got a positive and negative end, and we can see it compared to an ammonia molecule that is a totally different molecule, but it is still capable of forming one of these hydrogen bonds. It's just a weak interaction between the negative end of one polar molecule and the positive end of another polar molecule. They're two different molecules, and even though that they have a hydrogen bond holding them close together, this is still an ammonia. That's still a water molecule. We haven't made anything new. So hydrogen bonds and hydrophobic interactions are weak interactions between molecules. If we were to put these things in a pot of boiling water, these molecular bonds would be the first to break, and then we would get down to the chemical bonds. So chemical bonds much stronger than molecular bonds. They also make new substances. We said molecular bonds don't. So the next few slides take us into unique properties of water. And now that we've set up that water molecules are polar, and we've explained why, like what a polar molecule means, this polarity of the water molecule is going to be responsible for several unique characteristics that we see in water. One of those unique characteristics I mentioned is the fact that water is the only thing that we know of that is less dense in its solid state than in its liquid state. I'll go back and forth between these two pictures. You can see in the picture, all that we're looking at is water molecules. So there's no different substance between ice and water. still the same number of hydrogens and oxygens.
One's closer impacted than the other.
Say that one more time.
One's closer like together than the other.
Right. So what's different here when I go back and forth between these two pictures is that in liquid water there are fewer hydrogen bonds. Now, getting into like a physics part of this class, the temperature really is just a measurement of how fast these molecules are jiggling around. And if the temperature is high enough, obviously liquid water has to exist in a certain range of temperatures. If it's too cold, then the molecules stop moving around. And if it's too hot, they're so far away that they don't form any of those hydrogen bonds in the first place. So liquid water is kind of in this range where molecules are jiggling around and they're constantly breaking and reforming these hydrogen bonds. It's what allows water, as those hydrogen bonds are constantly breaking and reforming, to take any shape of the containers that it's in. That's the only water I had, and if I were to pour it out on the table, we would see the water take the shape of the surface of the table. Those hydrogen bonds constantly breaking and reforming allows water to adjust its shape all the time. Solid water or ice doesn't change its shape because each molecule is locked in this lattice of the maximum number of hydrogen bonds. Every end of these water molecules are completely locked to the other end of a water molecule. So negative ends of a water molecule are attached to the positive ends of an adjacent water molecule. And what's unique about this is that the addition of hydrogen bonds, we can see how it evenly spaces out these water molecules from one another. So in liquid water, like was mentioned, each molecule can end up being closer together because there isn't the maximum number of hydrogen bonds to kind of separate those molecules equally from one another. So the only difference, like we said, between ice and water is the number of hydrogen bonds. There are more hydrogen bonds in ice, and that's, like we said, what separates those molecules, making it less dense.